This chapter has everything to do with enzymes and reaction kinetics. So we recently finished off a couple of chapters on proteins and protein folding. Enzymes are a type of protein and they help catalyze different reactions. So this is something that we should focus on, and it's something that does have a bit of modeling with it involved with it, for example with the Michaelis-Menten equations. But what's most important is that most of you have taken a biochemistry course already, and so this will act as more of a review of the different types of concepts from an introductory biochem course leading into the models that we use and different steady states. So in this lecture, the first thing we want to talk about is a recap, because there were so many different concepts covered in the previous lectures that I do want to cover just a couple of the most important things that you should take home from the previous lessons. Then I'll jump into the main lecture, which involves an introduction to enzymes, then a full overview of enzyme kinetics, and some steady states and examples of calculating the Michaelis-Menten equations. So this is supposed to be a quick recap, but there were quite a few topics that we talked about last time. There was an overview of protein folding, leading into secondary structures. Then we presented two main models for protein folding. It was the two-state model of the alpha helix, basically your helix to coil transitions, and that meant we could figure out the fractional helicity of a protein. Then we moved on to protein-like sequence design using HP models, and then summarized everything at the end using energy landscapes. So first of all, what makes proteins fold? Just as a quick summary, we learned that hydrophobic forces are going to help proteins fold into compact conformations. So we want to focus on the hydrophobic core that is created. So that means polar amino acid side chains are going to want to be, non-polar amino acid side chains are going to want to be grouped in the middle where they're in contact with one another. Once they folded into a specific conformation or configuration, your hydrogen bonds are going to help stabilize your protein's folded shape. And how can we ensure proper folding? We have chaperone proteins. So they assist in the covalent folding or unfolding of different macromolecules. And they go in, for example, into the chaperone protein, get folded properly, and then get ejected out as a perfect protein that is functionable. Once we have a secondary structure, we learned that there are two common secondary structures of proteins. You have your alpha helix over here and your beta sheet. And the only differences between the two are the way the hydrogen bonds form between the NH and CO groups within the polypeptide backbone. So when we discussed this, we first went through a Ramachandran plot of which ones are more common. Remember, beta sheets had that big blob at the top left corner with different phi and psi angles, so that was the most common and the most abundant, whereas there were left and right-handed alpha helices that were possible. Now, looking at this, we have the coil to helix transition in a polypeptide chain. So this is when we introduced our first model, and basically it means your individual monomers or amino acids can be in a coil state, or after some hydrogen bonding become a helical shape. So there's going to be greater entropy here because there's a lot of free form shapes, and there's going to be a much smaller value of entropy over here because there's more order. So when we're modeling the alpha helix, one of the key things to remember is that your first monomer is going to bind to, or hydrogen bond with, your J plus fourth, or the fifth monomer. So you have 1, 2, 3, 4, 5. your first and your fifth monomers are going to hydrogen bond together, and that starts a cascade effect. So we did this, we did an analysis using a whole bunch of examples. So here, remember this, we had a protein coil, and slowly it starts to form a hydrogen bond. It forms the first hydrogen bond, which requires the most energy, and then a cascade effect occurs where the second one preferably binds to the 6th monomer, the third to the 7th, and the fourth to the 8th. And the reason why is because once I form my first hydrogen bond, every monomer 2, 3, and 4 within that ring is forced into that ring structure or that helix structure. They're still considered coil states, which means they want to be free to move, but they can't because they're essentially locked into place by this 1,5 connection. Therefore, it's not preferable for them to stay in this conformation. They do want to hydrogen bond with another monomer above them. So then you have a cascade effect where each additional hydrogen bond takes away one extra monomer until none are left. So then we tested ourselves to see whether some of these sequences are possible or impossible, and our end result is that if you have one hydrogen bond that's already started, everything else is going to want to hydrogen bond until everything is bound, except for the uppermost level of your helical structure. So if you have a 1,5 hydrogen bond, a 2,6, and a 4,8 hydrogen bonds, but the 3,7 is missing, then this sequence is highly improbable because the 3,7 monomers are absolutely stuck in place when they want to be free. So these are highly energetically unfavorable. So being able to explain why something cannot be is very important, especially when it comes to a test. So then we called this model with the helix coil transitions, we called it the Lifson-Roig model, which basically is a step above a graphical representation. It now assigns a statistical weight to the polypeptide's residues. So every residue can be either in a helical H or coil C state. So we assign a statistical weight to each one so that we can predict the fraction of the polypeptide that's in a helical or a coil state. So the first thing we did was we gave C a weight of 1 and H a weight of S. And that meant that we could use the binomial function to find all the possible sequences for a dimer. So something like this. Next, we figured out that we could find the probability of each sequence by dividing its individual multiplicity. So if I wanted the probability of getting the CC sequence, I divide the multiplicity of CC, or the probability of that, by the total multiplicity, which is calculated by your binomial expansion or your partition function, depending on how many monomers or subunits you have. So if you have 10 subunits, you'd have 1 plus S to the 10, or Z10 as a partition function. So I won't cover partition functions again. You can go through the previous lectures to go through that, but there are multiple ways of getting this denominator. And at the end of it, we were able to define an equation for fractional helicity, which is the ratio of helical states to the total number of monomers in the sequence. So therefore, we can get a percentage or a fraction of H's within the whole polypeptide. We then took a look at a couple more parameters. We wanted to figure out what the parameter S meant. So we introduced sigma, which I won't explain again, but it's very important to understand. So if sigma is equal to 1, the fractional helicity is given as S over 1 plus S. So we consider S the relative weight of H. And that means S can be calculated by the weight of H divided by the weight of C, because C was our normalization factor. So then you can get something along these lines, and note that the probability of H can also be rewritten as this term, which means we can relate it to temperature and entropy. So to summarize all of this, what we've discovered is that using the Lifson-Roig model, we can assign statistical weights to a helix or coil state. We can figure out the probability of getting each sequence, and we can also find the probability or the fractional helicity of that entire protein sequence. Once we figure out that, we'll be able to graph it. And once you graph it, because you know that the parameter S is related to temperature, it means we can also relate that parameter to entropy. So we ended off here where from the Lifson-Roig model, we found that sure, we have a coil state and we have a helical state. But the one important lesson from the Lifson-Roig model is the degeneracy. Over here, or the multiplicity, over here with the coil state, you can form so many different configurations or shapes. So the multiplicity of that denatured protein is way higher than 1. But if you want just one protein that has a particular function, then that protein needs to be formed properly, and therefore it means there can only be one possible shape for that protein. So when we consider a protein-like sequence, it means that you must analyze your entire sequence so that you can get a degeneracy of 1. So that's something we looked at using the HP model from compact random walks. So looking at this, the first thing I want to go through, a lot of students had questions about this before, about where this compact random walk came from and why it's like this. So remember when we took a look at the hydrophobic forces that would kind of fold the protein into its shape, and remember how all of the nonpolar amino acids were clumped into the middle? Essentially, the idea is that you're going to force or fold your protein into its most compact shape. So if you consider every amino acid or every residue in your polypeptide sequence to take up one space in a lattice structure, there are two assumptions we have to make. Number one, every single spot in the lattice structure is taken up fully. And number two, there is no overlap between the connection of those sequences. So that means we have to come up with different ways that the protein can fold in on itself and have a different structure. Then we can look at the different structures to see which one is the most or the least favorable. So that's the whole idea behind the HP models. So to start with the HP model, remember we're trying to simplify things as much as possible. We can consider each residue to be polar or nonpolar. So we moved on to something called the 6-mer toy model, or the HP model. So keep in mind they're both interchangeable. The HP model usually refers to a 6-mer toy model, where for a 2D 2x3 lattice structure, you have a whole bunch of different sequences, but when you try and connect the dots essentially to form a fully connected polypeptide sequence of 6 amino acids, you can only come up with 3 main shapes. You can either go all around, so that forms this U or pie shape. You can go back and forth like this, which forms your S shape. So that means you can go this way or this way as well. But no matter what and how you manipulate it in 3D space, you still end up getting that S-shaped structure. Then you have one that just goes all the way around, starting from the middle. So I could start here and go around. I could have started here and went around this way. But no matter what, it's still a G shape. There is no other shape that I can make from the 6-mer toy model. I can't go from 1 to 4 and then wrap around here because it crosses the 1 again. So there's only 3 shapes that I can create. So the next thing is to take a look at each of these models for one sequence to see if it's preferable or not. And so when we question you on an assignment or a test, we won't make you go through something like this where you have to figure out all the possible sequences. We'll be giving you a sequence and trying to get you to evaluate it by tacking on how many energy penalties there are. So for example, if I gave you this sequence at the top, which would be, if you use red for nonpolar and white for polar, you would have nonpolar, polar, so you would have HP, HP, HP. This would be an HP, HP, HP sequence. So if I assign a sequence to you, I expect you to be able to draw this out like this in the 3 different shapes that are the compact structures. And then I'd expect you to be able to tack on or draw in dotted lines the energy penalties or the non-preferred interactions, hydrophobic interactions experienced by each of these shapes. So here you have 3 unwanted interactions, 2 and another 2. So a total of 7 energy penalties for each of these. So at the end of it, when you're trying to figure out which sequence is more protein-like, is there any sequence here that's preferred? Probably not. So here, this would not be a protein-like sequence. Your HP, HP, HP sequence has 3 different conformations all at the same energies. Its multiplicity or degeneracy, which means we've now accounted for energy. Your degeneracy is equal to 3, which means it's not equal to 1, which means this protein cannot fold into a preferred shape. And that means that it is not a protein-like sequence. Whereas if you go to the other one, you have 3 different shapes. This seems to have the least energy penalties. It's the only one out of the 3 shapes that has this energy penalty. Therefore, this is a protein-like sequence. So this would be P, H, P, P, H, P. That would be a protein-like sequence because it has a degeneracy of 1. So what that means is the lowest energy penalty model is going to have a multiplicity of 1. So then, as practice, I wanted you to go through and try and come up with all the different sequences that are protein-like, and you should be able to end up with these 3 numbers. So that means evaluating a whole bunch of different sequences, and that's good practice. Another way of practicing is to just start assigning different sequences of proteins. You can just assign yourself HP, P, 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 something like that, and see if it's a protein-like sequence or not. So then at the very end, we took a look at 2 competing views of protein folding and then energy landscapes. So with the framework model, you can see here that an unfolded protein gets folded into its secondary structure through local interactions before the chain collapses. But with the collapse model, hydrophobic or external forces are going to drive protein folding, making it collapse first and then form a secondary structure. So those are the 2 competing views, and it would be good to know them if there's multiple choice questions, for example. Next, we have your energy landscapes. So out of the 4 of those, remember we said the golf course model was the worst model because it means for a protein to find its proper folding configuration, it'd have to test out all of these different conformations equally at random until it hits the perfect one with the lowest energy. Usually you want a sort of energy landscape, whether it's smooth and rough, or just smooth. You want to have it cascade or have a flow down an energy landscape. The more of a hill you have, the more the protein can adjust or reconfigure itself so that it becomes a more and more preferred structure. So that gives a good 20-minute recap of protein folding. Now we want to go through an introduction to enzymes. So I want to go through what are enzymes, what are activation energies, and how do enzymes work. So as a quick overview of enzymes, an enzyme helps to speed up energetically favorable reactions. So it helps to produce disorder in the universe. Biologically speaking, what enzymes do is to help lower the barriers that block chemical reactions. And they do that by lowering something called an activation energy, which we'll discuss in the next slide. An important thing to know is that enzymes do have an active site that binds to substrate molecules. We're going to take a look at this in detail in a bit. But once this binding occurs, it reduces the activation energy of a chemical reaction, and therefore an enzyme acts as a catalyst to a reaction. So on to the activation energy. Every reaction has a reactant and a product. So the reactant here has to collect enough energy to surpass some sort of barrier, basically an activation energy, in order to reach the product. But if you have a catalyst, what it's going to do, or an enzyme, what it's going to do is lower this barrier. It lowers the barrier so it's easier and faster for your reactant to reach this energy level to proceed with the reaction. So when we take a look at catalysts and how they lower activation energy, at any given instance, a population of identical substrate molecules will have a range of energies like we see on this graph. These varying energies come from collisions with surrounding molecules. So for a molecule to undergo a chemical reaction, the energy of the molecule must surpass the activation energy for that reaction. So the required energy is kind of highlighted here in red. So for most biological reactions, we can kind of see here the number of molecules that actually reach this energy naturally is very, very little. So that means we usually need enzyme catalysis in order to go through a whole bunch of biological reactions. So in the red shaded area, that's when reactions start to happen, and that's where the required activation energy is located. So there are a few things we can do to increase the number of molecules with sufficient energy to overcome this activation energy. So in contrast to enzyme catalysis, this effect is non-selective. So basically, in terms of this, I think I want to mention, if I want to speed up a reaction, do I care about temperature? And is it different than whether I use a catalyst or not? So in contrast to enzyme catalysis, increasing the temperature is non-selective, meaning it speeds up all reactions. This is super important because it is different than an enzyme. An enzyme is going to be specific to a reaction, and it's going to speed up that reaction only, and it's going to be selective. So temperature is non-selective speeding of reactions, whereas catalysts are going to be selective speeding of reactions. So looking at this, we just want to go through a few analogies with different types of enzyme catalysis. And this is a modeling course, so that's why I'm presenting a whole bunch of different analogies for different ways we can model enzyme catalysis. So we can represent enzyme catalysis using balls in a lake, where they have to crash through and surpass some sort of barrier in order to overflow onto a waterbed. So if we do have a catalyzed reaction, we'll be able to lower this barrier and allow a flowing stream to go through. So this one's a fairly basic model. Next, we have an analogy with boxes. So this takes a bit of getting used to, but you have this box with 1, 2, 3, 4 removable barriers, and each wall represents a different energy barrier, leading to a different reaction pathway. So we'll see a little bit more of that pathway in the next slide. But basically, you have some ball in the middle and some water, and you have to raise the energy so it knocks down certain walls. So if the water level or the energy is raised to a certain amount, then a catalyzed reaction should be able to knock down the barrier and allow the ball to flow through pathway number 1. Same as if it raises it up to energy level 2, it's going to knock this wall down and go through reaction number 2. So, do you think this is a good or a bad analogy? So the assumption is that once you reach a specific wall's activation energy, it's going to spill over and the reaction progresses along that pathway. But if number 1 is the lowest, how will you ever get to 2, 3, and 4? So this isn't the greatest analogy unless we can assume the energy levels jump from one level to another, but most energy level increases are gradual. So in this case, I would say it's a good try at an analogy or a model for enzyme catalysis, but it's not the greatest. So why don't we look at a better analogy with branches? We can represent different enzyme catalysis using a branching river with a set of yellow barrier dams. So this is just taken from a couple of different textbooks. It's an expansion on the previous analogy, but now the assumption is that we've continued the chain of reactions. So maybe there's only 2 or 3 options for each box or river dam that we've looked at. So for this one, we have 3 possible pathways. Depending on which enzyme is used to catalyze the reaction and where that energy level is, different walls will be knocked down and different pathways initiated. So if your enzyme is added here and knocks down this barrier and goes through this pathway on the left, then another enzyme comes in and knocks down another barrier and continues here, you get this product. So at the end of it, through various different pathways, you could potentially get a whole bunch of different products. So how do enzymes work? Every enzyme has an active site, which binds to substrates. So looking over here, we have an active site, and your substrate can bind to that enzyme. And then it goes through catalysis. So the reaction occurs and produces an enzyme product complex over here, and then your product is released. Your enzyme is returned to its original state, and you can continue making more and more of these products. So we now want to go through the main part of this lecture, which has to do with reaction rates of enzyme catalysis. So this is a pretty short chapter because you've probably learned all of this before. But we're going to go through an overview of different reactions, the Michaelis-Menten model, which is the most important model for this whole chapter, and some equilibrium and steady state approximations. So assumedly, we would have a reaction rate for a simple reaction where A plus B goes to C. So without any additional help, molecules usually collide with each other due to random walk. We can then calculate the reaction rate for these collisions. This is usually called the collision rate. So we calculate R is equal to negative dA over dt. dA over dt is the rate at which we are losing the concentration of our reactant A. And because there is no C initially, because we're starting with just reactants only, we know that the reaction rate is fully dependent on A and B initially. But what happens as the reaction progresses and C is made? So after the products are formed, I want to also mention that this dA over dt is the rate that you're losing A, but it's also going to be proportional to the rate that you're losing B, because A plus B goes to C. So now we take a look at products that are formed. What happens after a whole bunch of product C's are formed after a reaction? First of all, let's define two types of reactions. We have a forward reaction where A plus B goes to C, and that requires collisions. We introduce a rate constant K for a forward reaction, so K plus. We also have a backwards reaction where C goes to A plus B, which doesn't require any collisions whatsoever, so we introduce rate constants K negative for the backward reaction. You end up having a rate constant being equal to the forward reaction of A and B minus the backward reaction of C, so that we can calculate the rate of overall reaction. So essentially, you're trying to calculate the number of products formed minus the reverse reaction to figure out how much C is actually being produced at any point in time. So then we want to take a look at the free energy of a system. I know in the past that I haven't really defined whether to use F or G for assignments, because whether you use F or G is usually ambiguous to the field that you're studying. So in biochemistry, the free energy, G, is the quantity that expresses the disorder of the universe. The value of G only matters when the system undergoes change. So here we're taking a look at Gibbs free energy. And that's all we're going to take a look at in terms of thermodynamics for enzyme kinetics. So the change in G is critical in determining whether a reaction is energetically favorable or unfavorable. And remember, we always want to have a negative change in G if you want to have a favorable reaction. So to explain this, there's a really good graph over here, or schematic. For an energetically favorable reaction where Y goes to X, the free energy of Y is going to be greater than the free energy of X. Therefore, the greater the free energy, the delta G is going to be negative, and the disorder of the universe increases when Y is converted to X. So it has a direct effect on entropy. Then you have an energetically unfavorable reaction. If the reaction X to Y occurred, delta G would have to be positive, and that means the universe would have to become more ordered. In that case, that's usually not favorable, which means it has to be coupled to a second favorable reaction that supplies more energy for it to surpass this barrier. So unfavorable reactions can still work if they're coupled with a favorable reaction. So reaction coupling allows an overly negative delta G from an energetically favorable reaction to help drive an energetically unfavorable reaction. So when you couple the two reactions together, the overall delta G is negative, but when you take a look separately, this one's positive and this one's very negative. So if there's a question that asks you whether unfavorable reactions can still occur and how, then using a description related to reaction coupling would help you answer that. Next, we want to take a look at the reaction coordinate. So catalytic reactions can be represented by using an energy versus reaction coordinate graph. So something like this. And here, for the forward reaction, I have to supply a delta G plus for the forward reaction. That means I need to supply an activation energy of this in order for it to proceed to form the product. For the backwards reaction, I need to surpass a much greater activation barrier. So it's delta G negative, that's the activation energy needed to go backwards. So I want to mention also that as A and B squeeze together, the free energy is going to increase. But once, and that makes sense, but once the dimerization starts to happen, your G is going to decrease and more product is made. So the next thing I want to talk about is the Arrhenius equation. In the late 1800s, Arrhenius created an equation to describe the relationship between temperature and reaction rate. So we ended up with this equation, where K, the rate constant, is equal to A, the Arrhenius constant, times e to the power of negative activation energy, EA, divided by RT. And we want to be able to tie this back to free energy and the Boltzmann's constant. So we're going to assume that delta G is much greater than kBT, and therefore we can say K is proportional to e to the power of this term. So we assume here that delta G is going to be the activation energy in each of the forwards and backwards reactions. Next, I want to discuss what happens after an enzyme helps a reaction. So using that same diagram, we have, or a similar diagram, this should really be flat. So you have an energy level here that it starts out at. With the help of an enzyme, the activation energy is lowered so that it's easier for the reaction to proceed, and therefore the backwards and the forwards activation energies are reduced. So now we're moving on to a conceptual model of an enzyme. So what this is saying is that there are 4 steps to enzyme catalysis, and we're going to go through this in detail, and it would be important for you to understand this and what's happening here. So your enzyme E over here, which is that box, has a binding site, this circular area, and it has a certain distribution of charges. To match perfectly, your substrate S has to deform, or both of them have to deform, so that your bonds can come together close to a breaking point. So essentially they have to bend and fit perfectly with each other. Step 3, so when they bind together, they form the ES state. In step 3, from the ES state, the stretched bond can break and give rise to a so-called EP complex. So all this is saying is that there's a certain reaction occurring here where the enzyme has now deformed your substrate, and that deformation is going to break or weaken the bonds within your substrate. And after that, you have an EP complex that's created. New bonds are formed to stabilize this new product, P, in its new shape. And because the new shape is not a perfect fit, it unbinds, creates a product, and your E goes back to normal. So looking at the free energy landscape, we start off with a substrate, and this is the simplest one. We're going to look at a few different energy landscapes and then superimpose them on one another. So the free energy landscape here, I start with a substrate, S, and unassisted through some sort of activation energy after it passes through its barrier, it forms a product. So this is an S to P reaction. Next, we have the second step where the ES complex is created and you have these weak bonds in between that are holding it together. So then these weak bonds are going to help lower the energy in the bond to make the whole complex a little bit more comfortable and more stable. So it helps lower the energy required to keep it in place. the third step is the creation of new bonds. The creation of new bonds is going to increase the free energy input into the system. It's going to have a deformation of energy due to the ENS complex. And at the end, when you add everything together, you end up with a free energy landscape that looks like this. So altogether, we have these diagrams. We start with a substrate to a product, an unassisted reaction that looks like this. There's going to be some sort of interaction between your enzyme and substrate that helps lower the activation energy, but then the deformation of your substrate turning into the product with the creation of new bonds is going to offset that a little bit. So you superimpose them all and you end up with a curve in the red. And that gives you a sum of all of the above that has the enzyme plus substrate creating an ES complex, and then that gets split up into an enzyme plus a product. So keep in mind, this last part is not a reversible reaction. So that's important to remember. This part is reversible. Just in case the enzyme and substrate come together towards the complex and aren't a great match, they can go back to their enzyme and substrate separately. But if they form a product, the product will no longer fit into the enzyme, so therefore you can't go back to the enzyme substrate complex or reform the bonds that have been broken. So here's just another example or another diagram of the different types of events that are occurring through the reaction coordinate. So first there's binding that happens. It forms an ES complex. Catalysis occurs and you have a transition state, and then you end up with the enzyme product complex, which then spits out the product because it's no longer compatible with the shape of the enzyme. So looking at enzyme kinetics, when we look at a catalyzed reaction, we have to determine what kind of rate constants there are. So let's assign these three values as our rate constants. We have K plus for the forward reaction, K minus for the backwards reaction from ES to E plus S, and we have a positive rate constant because it forces every ES complex to go towards E and P. There is no reversible process for this one. So another thing to remember is this is called the binding event, and this is called the catalysis event. So if there are multiple choice questions or conceptual questions that ask which processes are reversible or non-reversible, do know the difference between these two. So our job now is to figure out what this Michaelis-Menten model is. So I have this equation here. I want to solve for the reaction rate. V equals dP over dt. That means how much product am I getting per unit of time? So the first thing is, why don't we just solve for the rate for each of these? If I have the rate of E with respect to T, it's going to be how many different ways I can remove or add E. So I can remove E by taking it away and putting it towards ES. That means I have to do negative K plus times E times S. That means it's going to give me the concentration of E times the concentration of S times the forward reaction rate, and that is going to take away the amount of E that I have in the system. But if ES goes through a backwards reaction, it's going to add to the number of E's in the system. So we have to add K minus ES. And of course, I can get more enzymes if ES goes forward and produces E. So then I do plus R times ES to get to E. So this is the full reaction equation for just the enzyme. If I'm looking at the substrate, I do the exact same thing. It would be good to try and derive these yourselves as well as practice. But at the end of it, you have dP over dt. This is the most important one. I want to find out how much product is being created per unit of time. So the only way to get that is for ES to go through the forward reaction at a rate of R and go to P. So it's R times ES. And that is the rate of the reaction that we look at. So we then see if there are any conditions. And we have this equation here and these additional rate equations. The initial conditions that we want to look at are as follows. When t is equal to 0 at the beginning, we have all of these values. So we can assume everything is 0. There's also conservation laws which say that initially E is going to be equal to ES plus E. And initially, S is going to be equal to ES plus S plus P. So try and think about why these are, because it might show up on an assignment question. So for concentrations, we now want to take a look at what happens when we change the concentrations over time. So, or not when we change it, we want to see how the concentration changes over time through a reaction. So we want to take a look at the enzyme, the substrate, the complex, and the product. So some trends, taking a look at this graph, my product is going to increase over time, which makes sense because I want more product. The substrate is going to decrease over time, and that makes sense too because more product is being formed from the substrate. But then I take a look at the enzyme substrate complex and the amount of enzyme that's available. The enzyme that's available is going to decrease very quickly because a lot of it's going to be used up for the enzyme substrate complex. But because the enzyme substrate complex is then going to release a product after its transition or after its catalysis, your enzyme gets released again and is then free to repeat its use in the enzyme substrate complex. So over here, we reach what's called a steady state. So in the middle, when things aren't changing too much, we call this a steady state and say that the concentration of the enzyme substrate complex is almost constant since R is very small. It takes a while to get to 0, so we can see that in the middle over here, there's going to be a plateau for ES, and it will take a very, very long time for it to get to 0. It'll go off the page and continue for quite a while. So where exactly, or what exactly can we get from this graph with relation to reaction rates? We can then use the Michaelis-Menten model to define the reaction rate once more. So now we're now switching to something else. We call Km the Michaelis constant. We call V the reaction rate, which was equal to dP over dt. And we call V max the maximum reaction velocity. So then, if I want to redefine everything, I can redefine it as this. V max times S, which is your R times S, for example, divided by Km plus S. We're going to derive this in a bit. So for the Michaelis-Menten model, the following two approximations are usually made. You have the equilibrium approximation and the steady state approximation. The equilibrium approximation states that the forward reaction rate, or the catalytic reaction constant, is approximately 0 because it's very small to begin with. Using R equals 0, let's try to find an equation that will allow us to graph reaction velocity versus substrate concentration. the steady state approximation states that the change in ES over time is approximately 0, because remember, it plateaued, which means that when you have a plateau, your slope is pretty much 0, because your value doesn't change and it's a constant. So we want to now derive the equation for Michaelis-Menten's model. We have to use the two approximations we've just covered for the Michaelis-Menten model and derive an equation for reaction velocity V equals dP over dt using the Michaelis constant Km, and then we want to plot it on a V versus S graph. So we want to just quickly move over to a slide that I've shown before over here. We know we want to find dP over dt, which we previously defined as R times ES. Well, one of the things we can do is get the rate constants in. So we can get K plus, K negative. We can get them all in by using dE over dt. And we can tie ES and E together by using one of the conservation laws. So why don't we start there? So step number 1, we're going to group the constants together in a rate equation. So we have 0 equals to dE over dt, which was from our conservation equations or our different laws. It's equal to this expression, which we know. The other thing we know is that we have an equilibrium approximation where this R is equal to 0. So that means we can turn that into 0. And we can rearrange and solve for the rate constants K. So we get K plus over K negative is equal to ES over E times S. Step 2, we want to solve for E, and we're going to use the initial concentration of the enzyme as ES plus E in order to find ES. So we redefine everything so that we solve for E, and we get ES K negative divided by S K plus. But we also know that E naught is equal to ES plus E. So rearranging that and plugging it in, we can factor out the ES and get 1 plus K negative over K plus S. So solving for ES, we get this term. So what do we do next? We now have ES, but we also know that dP over dt was equal to R times ES. So why don't we plug it in directly? So we have V equals to dP over dt equals to RES. We just solve for ES, so we replace this for the whole thing. We also know that KD is equal to K negative over K plus. So that's called a dissociation constant for ES, so we can rewrite it as, so this can be 1 plus KD over S, but keep in mind the maximum velocity is also going to be R times E naught. So I can replace this whole thing with Vmax. So I now have Vmax over 1 plus KD over S, or if I rework this, I get Vmax times S divided by KD plus S, especially if I factor out the the 1 over S over here. So now the steady state approximation implies that dES over dt is approximately equal to 0. So why don't we choose another rate equation to analyze? We can actually set this equal to 0, solve for this, replace the K negative in the equilibrium approximation by K negative plus R, so we get this term. But the other thing we know is that Km is equal to K negative plus R over K plus. So that's why we kind of rearranged everything like this. We rearranged it so that we can get to the Michaelis constant. And once we define the Michaelis constant, we can then say V is equal to V max S over Km plus S. So that's a more mathematical modeling derivation for your Michaelis-Menten model. So the last step was trying to graph this. So step 6, we wanted to graph the curve. Km is defined as the substrate concentration at which the reaction rate is half of the maximum velocity. So Km is the concentration at which 1/2 Vmax is reached, whereas Vmax is all the way over here. So if you were ever asked to define the Michaelis constant conceptually, that would be how you would define it with the help of a graph. So then we now present the Michaelis-Menten equation as this. So we've seen this before, we were able to derive it. V is equal to the change in product over time, which is equal to V max S over Km plus S. We can determine Km and Vmax from one of these plots. But I want to ask what happens if Km increases and what causes this. So when Km increases, that means that a higher concentration of substrate is required to reach half of the maximum reaction speed. So that means the larger the Km, the more substrate is required to reach this point here. The Michaelis constant is a measure of binding affinity of the enzyme for the substrate. So the larger the Km, the smaller and weaker the binding affinity, because that means more substrate is required. The smaller the Km, so the closer it gets to the origin of the graph, the stronger the binding affinity, because it's going to bind directly to your substrate and start the reaction right away. So if a question asks you what Km would be if you have a stronger or weaker binding affinity, you should be able to answer this as well. The next thing I want to talk about is something called the Lineweaver-Burk plot. So sometimes you want a linear graph to make things easier. So you can take the reciprocal of the Michaelis-Menten equation to get a linear graph. And you get an equation like this, which means I get to easily look at the intercepts and get 1 over Vmax, and the slope is equal to Km over Vmax. The intercept here is -1 over Km. So you can derive these yourself, but most importantly, by fitting data to this curve, you can easily find information on Km, Vmax, etc. So some additional comments I want to present to wrap this up. The Michaelis-Menten mechanism must apply both equilibrium and steady state approximations. So for an assignment question, I might ask you to re-explain the equilibrium and steady state approximations in your own words so that you can try and discuss why it's required. So the Michaelis-Menten mechanism applies to many different kinds of enzymes, including lysozyme, and that's just an example. The formation of the enzyme substrate complex is very fast and reversible, which means it can be spontaneous. But the formation of a product is going to be slow and irreversible. So those are two key differences to remember, especially for conceptual questions. So what did we learn today? We learned that enzymes help to lower activation energy barriers. It helps push forward reactions, and it allows us to know the difference of effect between temperature and catalysis. We learned to understand the energy landscape of enzyme reactions, so definitely look at the graphs of enzymes, substrates, and products to see how those graphs work and why they're like that. And definitely know what types of conformational changes are occurring in each of the 4 steps. The last thing we learned about was the most important model for enzyme kinetics, the Michaelis-Menten mechanism. like I said before, understand the two types of approximations used and know how to read the graphs and get valuable data from it. So there might be some questions where you're asked to label a graph, or you'll be asked to try and figure out the Vmax or the Michaelis constants just by looking at the graph.